Okay, you have made it to the very last lecture of the semester. So we have talked about how we made the RNA, the messenger RNA, from the DNA transcript. So that was transcription. We talked about how we processed that RNA before it came into the nucleus. We talked about how we matched the tRNA with its correct amino acid. We talked about how we put together the ribosome. Now we're ready to actually start translation. So translation is going to start at the start codon, AUG. So that AUG is going to bind to the small ribosomal subunit and to the tRNA. So those are all going to bind together, the initiator tRNA with the mRNA and the small subunit. The small subunit moves along the RNA until it reaches the start codon, and initiation factors are going to bring in the large subunit, and that makes the completed translation initiation complex. Then during elongation, so we start here, the amino acid comes in, the amino acid is brought in on the tRNA into the A site, so then the A site and the P site are both filled. The E site amino acid forms a peptide bond with the A site amino acid. Then the whole thing shuffles down, so it's just going to scoot down, and then that amino acid from the P site moves into the E site and goes off. It can go find a new amino acid and keep working. The tRNA from the A site moves down to the P site, and the next amino acid can move into the A site. Then that's going to come into the A site. They're going to form a peptide bond between these two amino acids. The growing chain is transferred over. The whole thing shuffles down, just again and again and again. It does take energy to form that peptide bond, and it does take energy to shift the whole thing down. So it takes a little bit of energy in the form of GTP, which is a little different from ATP, but we're not going to talk about that. So it just takes a little bit of energy to make the whole thing happen, which is not surprising. Anytime you build anything, any type of anabolic reaction is going to take energy. Translation moves in the 5' to 3' direction. Just keep bringing in the next amino acid. Your tRNA brings in the next amino acid. tRNA brings in the next amino acid. It just moves down codon by codon. Each cycle takes less than 1/10 of a second in bacteria. So that means every second a bacterial cell could add 10 amino acids to that growing polypeptide. Elongation is going to keep going on until a stop codon in the mRNA reaches the A site. You remember those 3 stop codons we talked about, UAG, UAA, or UGA. When a stop codon gets into the A site, the A site will put in a release factor protein. So instead of putting in a tRNA, you just put in a release factor. The release factor adds a water molecule instead of an amino acid. That makes the whole thing just disassembled. That RNA could go and be read again and again and again by different polypeptides until it gets broken down. That mRNA could be read again and again by different ribosomes to make new polypeptides until the RNA gets broken down. These ribosomal subunits can go attach back to each other or attach to other smaller large subunits, and so that can just keep that RNA is going to make a lot of proteins, not just the one. Translation is often not enough to make a functional protein, though. Afterwards, you often have to have post-translational modifications. So you're going to have modifications happen after translation. Those polypeptide chains can be modified, and often that modification is what helps it actually become active, or it can help it move to specific sites. So target it to go be put into a specific place. Here's one example of this polypeptide. Starts with a signal peptide. That signal peptide is going to signal this molecule SRP. SRP binds to the signal peptide and carries that signal peptide to the ER. So that is now going to turn this free ribosome into a bound ribosome. And so that bound ribosome is able to create this protein and put the protein right into the membrane where it needs to be. So that protein can be just deposited right into the ER, and the signal peptide is going to be cut off. It's not needed anymore. Then the ER then the ribosome can go off and the whole thing can happen again. Let's see. During synthesis, the polypeptide chain can start to fold. It can start to develop its secondary or even its tertiary structure during polypeptide formation. The genes itself are what determine primary structure. The genes are the primary structure, and the primary structure is what's going to determine its secondary and tertiary and quaternary shape. If you don't remember that, please go back and review that again. Post-translational modifications could be required before the protein can start doing its job in the cell. So those post-translational modifications really get that proper function in the protein. We do have free ribosomes and bound ribosomes. Free ribosomes in the cytosol and bound ribosomes attached to the ER. That is mostly as a result of the signal peptides bringing those ribosomes to be bound. The ribosomes themselves are identical and can easily switch between being free and bound. Polypeptide synthesis always begins in the cytosol, and it finishes in the cytosol unless there is that signal sequence. So that signal peptide is what causes things to be brought to the endoplasmic reticulum. Okay, we talked about that. Multiple ribosomes can translate a single RNA simultaneously. So you can have just ribosome after ribosome coming down, making lots of proteins at the same time. Polyribosomes let a cell make a lot of copies of that protein very quickly. Here you can see a bacterial cell that has ribosome, 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 all these ribosomes bound onto this RNA. You can't really see the RNA well, but it's this kind of string that goes through the middle of them. So that cell can respond to whatever change, whatever signal it got, and make a lot of proteins very fast to very quickly be able to accommodate whatever signal or change it got. So a bacterial cell has that streamlined transcription and translation occurring together, and then the protein can diffuse to where it needs to function. It happens very fast. In eukaryotes, the nuclear envelope separates transcription and translation, and you also have translational, pre-translational modifications before it comes out of the nucleus. Here's a fun little clip.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon, that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
Okay, so that's kind of overviews the whole process. Here's another little clip.
In translation, a cell reads an mRNA message and assembles a polypeptide accordingly. We can divide translation, the process of building a polypeptide, into 3 stages: initiation, elongation, and termination. Translation is initiated when the small ribosomal subunit binds to the leader at the 5' end of the mRNA molecule. The anti-codon of the initiator tRNA binds to the start codon, AUG. The initiator tRNA always bears the amino acid methionine. Proteins called initiation factors help bring the mRNA, the initiator tRNA, and the small ribosomal subunit together with the large ribosomal subunit to form an initiation complex. The initiator tRNA sits on the P site on the ribosome. Elongation of the polypeptide now occurs, with amino acids being added one by one. The codon in the A site of the ribosome pairs with the anti-codon of the appropriate tRNA molecule. Part of the ribosome catalyzes the formation of a peptide bond between the amino acid extending from the P site and the amino acid extending from the A site. The tRNA in the A site now translocates to the P site. The tRNA that was in the P site moves to the E site and exits from the ribosome. Meanwhile, a new tRNA brings its amino acid to the A site and the process is repeated. Three special base triplets, UAA, UAG, and UGA, do not code for amino acids, but instead act as stop codons, which terminate the process of translation. A protein called a release factor binds to the stop codon in the A site. It acts to free the completed polypeptide from the tRNA that is in the P site, and the translation assembly comes apart. The polypeptide becomes folded, modified, or combined with other polypeptides to form a functional protein.
Okay, so we've talked about how transcription occurs. We've talked about how translation occurs. This last thing we're going to talk about is mutations. So a mutation is when you change the genetic information of the cell, when you change the actual DNA. We're specifically going to talk about point mutations. Point mutations is a change where you only change one nucleotide in a gene. Sometimes that doesn't lead to any change because of that wobble of the genetic code. So you might end up making the same protein. Sometimes it can make a very big difference. Cystic fibrosis is a disease that is only the change in one nucleotide. Sickle cell is a change in one nucleotide. So here's the wild type, the typical shaped red blood cell. And in sickle cell, you have this red blood cell that's shaped like a sickle, so it doesn't carry your oxygen through your body as well. And instead of having a T, it has an A. So that one change here changes our RNA from being GAG to being GUG, which then changes from having glutamine in your normal hemoglobin to having valine in your sickle cell polypeptide. So by changing the DNA, you end up with a mutant RNA and a mutant protein. Sometimes that mutation might not do anything. It would just be a silent mutation. Sometimes that mutation does something harmful. So here, sickle cell is not able to carry oxygen as well, so that can be harmful. Sometimes it does something positive. So sickle cell is less likely to be infected with malaria, so that's a positive change. So if you are living somewhere where malaria is rampant, that's a positive change. That is an evolutionary benefit. But if you are living somewhere like up in the Alps where it's hard to get oxygen, that is going to be very fatal. So that would be a disease. Anytime that the effect is a phenotype that is adverse, that is harmful, this change is going to be called a genetic disorder or a hereditary disease. So a genetic disorder is something on your genes leads you to having a disordered function, or hereditary disease because your genes are inherited and it causes the disease. Point mutations within a gene can be divided into two categories. One are single nucleotide pair substitutions, and one is going to be insertions or deletions. These are both just going to be changing one changing one nucleotide in your DNA, which then is going to change one nucleotide in your RNA, which then could change the protein in your polypeptide. A nucleotide pair substitution is when one nucleotide gets replaced with another. So here is what the normal DNA strand should be, what the normal RNA strand should be, and what the normal protein should be. So in this nucleotide pair substitution, it can cause a silent mutation. A silent mutation means you have changed this CCG into a CCA. So now you have GGU instead of a C in your RNA, but you still put in the same still put in the same amino acid. You've got a glycine there. So you do not have any effect. That is a silent mutation, and that is silent because of the redundancy of the genetic code. Please go back and look at that chart to refresh your memory. The genetic code is redundant. So both this codon and this codon both give you the same amino acid. That's a silent mutation. There are missense mutations. Missense mutations still code for an amino acid, but they give you the wrong one. So if you had this mutation in your DNA, put a T instead of a C, you end up with AGC instead of GGC. You have serine. So that serine, if it functions similar, if the amino acid that's changed is similar to the amino acid that it was originally, then it might not cause a difference. But if you put in like a nonpolar amino acid where you had a polar amino acid or vice versa, or a charged amino acid where it used to be nonpolar, you can change that protein folding a lot. So this might cause anything from a small, slight change to a very large change in how that protein folds and how it interacts with its target molecule, how it's going to function in the cell. There's also nonsense mutations, which are going to create a stop codon early. So here, instead of having AAG in your RNA, you have UAG. So this T was changed to an A. You end up with UAG. That is a stop codon. So it starts transcription, starts translation, puts in a methionine, gets to the stop codon, and stops. The whole thing falls apart. So you do not make a functional protein. You can put that stop codon anywhere. It could be mutated anywhere, but you end up with stopping the translation of that protein before it's supposed to stop. So you end up with a non-functional protein almost all of the time. It's going to be incomplete and just not work. Then there are also insertions or deletions. So here is this DNA strand again, the wild type, typical, healthy version of the gene, the typical version of the RNA and the protein. A nucleotide incision, you put in an extra A, it's going to shift it. Or if you take one out, you're going to suddenly shift it. So instead of having this codon, this codon, this codon, by taking one out, you just shift the codons to this way, or you could shift them the other way. So you have a frameshift mutation. Does that make sense? You end up just those sets of 3s are suddenly off because you stuck in a 4 or a 2 somewhere. So you've had to realign your entire coding strand. That could put in an extra stop codon, so you might end up with a nonsense mutation. You could have an extensive missense mutation where you just end up with everything after that amino acid, everything after that nucleotide was taken out or put in, everything after that, the frame is shifted. So everything after that, you're getting weird amino acids. So insertions and deletions cause wildly crazy mutations, because who knows what kind of amino acid sequence you're suddenly dealing with, what kind of protein you could end up, or what kind of protein you could have stopped making just by taking in or putting out something. Spontaneous mutations can occur during errors in DNA replication, during recombination, or during repair. Mutagens are physical or chemical agents that can cause mutations. So something like UV radiation can cause mutations. Sunlight can cause mutations. It has radiation in it. Cigarette smoke, anything that's labeled as this causes cancer, it is a carcinogen. And most carcinogens are mutagens, and most mutagens are carcinogenic. So if it causes a change in your DNA, it's at risk of causing cancer because you are now allowing cells to grow in ways that they shouldn't be growing. Okay, so just reviewing, what is a gene? A gene is a discrete unit of inheritance. It is a region of a specific nucleotide sequence in a chromosome, and is a DNA sequence that codes for a specific polypeptide chain. So all of that, a gene is a region of DNA that can be expressed to produce a final functional product that is either a polypeptide or an RNA molecule. Okay, that's it. I will raise my glass, my little kitty mug. Cheers to you. You have finished the semester. I hope to see you back another semester. Best of luck, and you're always free to come back and see me, visit, and talk about whatever you may need. Even if you're not in my class anymore, I still love to see all the success you have in life. Bye.